the cellular processes that are going wrong and how we design drug targets to uh, to circumvent those systems that have gone wrong at a cellular level. Today, I'm going to take a step back and talk about how we design cancer treatment in the context of basic cell system at molecular biology level, right? So, everybody now, I think in this audience, knows about what's a central dogma of life, right? At a cellular level. And this applies right from virus to bacteria to organisms and up to mammalian systems, right? That we have a DNA where the genetic information is stored. DNA undergoes replication to increase in our copy numbers. Then we have transcription where DNA information is copied into RNA, and then we have translation where information is translated into proteins, right? And these proteins are the ones that carry out various functions. The drug targets that we talked about was further upstream from once the protein is synthesized, the complex interactions of these proteins with various different pathways. That's what we talked about last. Now, today we'll focus on this central principle of life, how things go wrong in this, and how we try to address these abnormalities with drug development and targeted systems. Right? So let's start um, with although it is very simple that um, central dogma DNA to RNA to protein, the entire system for human system or mammalian system takes place in the context of a cell. So it is divided spatially. Most of the DNA replication and transcription happens in the domain where nucleus is. mRNA gets transported out from the nucleus to cytoplasm. That's where it gets translated. And some proteins have intracellular functions. Some proteins get exported out of the cell system and have extracellular functions, right? All these tiny points uh, become important to note down when we reach the complexity of cancer initiation, cancer progression, and developing treatments to mitigate the cancer. We'll start slow and we'll reach the complexity by the end of the talk. Yeah. Anytime in between, if you have any questions, please start talking because I cannot see the audience. I cannot see myself also. So if you start talking, I will understand that somebody has a question. Yeah. So now let's start from DNA, the very first step of DNA replication. Now DNA just doesn't hang around in the system, right? It's a very long thread of deoxyribonucleic acids, and it is packaged so that it stays inside the nucleus system, right? So it is wrapped around histones. Histones get coiled over, form condensed chromatin, either heterochromatin or euchromatin, depending on whether it's actively transcribed or not. And these chromatin structures get further condensed into chromosomes, right? So all of us are diploid animals, and we have two copies of each of these chromosomes, right? So we have developed a karyotyping system where we have a chromosome spread. That's where the chromosomes structures and the counts we can see through various different systems. So this was the very first window in diagnosing cancer that was developed. Most of the cancers that first came into our understanding were blood related cancers, hematological cancers, right? And hematological cancers in karyotype, majority of them show various different translocations. So this became the very first technique to understand that something is going wrong with the genetic at the genetic level, at the DNA level, and that's what is leading to a cancer development. That was the very first window for us to enter into what's going wrong into cancer system, why the cancers are developed, right? So blood system, the blood cancers, hematological cancers were the ones that were discovered earlier. The reasons for those, the mechanism for those, and why it was accessible and easy. Karyotyping cytology technique exists more than 50 years in our system, and it was very easy to look at chromosome spreads, and most of the hematological cancers have major translocations, as you can see here, right? So these major translocations are very easy to spot at a macromolecular level. Yeah, chromosome is a macromolecule. It's a huge molecule, visible even at 4x 5x magnification under the microscope. And any abnormalities that are happening, very easy to spot for human eye using a simple microscopy technique. Right? So these became as landmark identifications that what type of translocation you have and what are the reasons you're developing particular hematological malignancies, blood-related cancers. Um, so most of these maps, as you can see, are already each particular blood cancer is mapped to a particular chromosome translocation, and the summary actually depicts quite beautifully that chromosome 21 gets translocated. Down syndrome, everybody knows, yeah, it's not a cancer related system, but it's still mapped here. Otherwise, the rest of them, most of them, you can see that they are blood related cancer, right? Lymphoma, leukemia, all these are types of leukemias. There is one sarcoma that we see, the two sarcomas that we see. Sarcomas are the cancers in muscle related systems, right? Rare diseases, but they are also initiated due to large chunks of chromosome translocating from one to another. So this was the very first window, and things are going wrong at a massive level, macro level, at micromolecular level, right? This part we already covered. So going further inside from chromosome level and how DNA replication, the central dogma, is superimposed on cell cycle. So we talked about in the last lecture how hyperproliferation is the fundamental reason cancers progress, right? And how various different therapies are targeting that hyperproliferation. Now, this is coming back to the basics of hyperproliferation. What are the different mechanisms that cancer employs to increase the proliferation cycle? Is the time increase? Is the frequency of the cycle increase? Right? So that's what we're going to look at, which directly relates to the DNA replication. Right? For every cycle a cell goes through, you have to double your DNA content. Right? So DNA replication is always tied with cell cycle. Now, all of us know the fundamental phases of cell cycle. A resting cell in this is an interphase. Interphase is when you have the tightly wound two pairs of chromosome. All the spreads, chromosome spreads that we saw in the previous slides are mostly from the interphase, right? Uh, so the cell starts building up to cell cycle, enters into G1 from interphase. Once the cell in G1, most of the DNA replication machinery is synthesized, and as the all the components that you need for DNA replication are ready, the cell enters into DNA replication phase. This is very short phase. That's where DNA gets replicated. Then the machinery uh, to build separation of chromosomes and separation of cells is built in G2. All the DNA damage response and correction of DNA damage happens in G2 after DNA replication. And once all the damage is corrected in G2, the cell, when the DNA is good to go, there is a checkpoint which the cell sends the signal to enter mitosis. Once mitosis is complete, cytokinesis happens and the cycle continues. Generally at this phase, cell can come back into interphase, right? So checkpoints generally are quite important so that all the errors that are happening during cell phase are corrected before the cell undergoes complete duplication and starts two copies of the cells. Any problems that happen at any of these checkpoints, the cell cycle is halted, and most of the times cell undergoes apoptosis, senescence and apoptosis, or apoptosis directly, right? So these checkpoints are at the interface of G1 to S1 to G2, right? So I had some checkpoint, right? So G1 to S to G2, and G2 to M. These are the various different states of chromosome, DNA, and cell membrane at various different points. So this picture talks about how DNA damage is corrected at various different points, right? So most of the checkpoints are guarding your entry into S phase and coming out of G2 into mitosis. Yeah, there are a couple of checks or spindle related check happen that during mitosis when two chromatids separate into two cell systems, right? We'll talk about various different DNA damage repair systems in further detail in next parts, right? So what are different DNA damage agents exist, and there are various different modes that are employed to repair that DNA damage, right? So this is to ensure that any kind of abnormality that happens into the DNA is corrected within time at appropriate times to preserve our genetic integrity, genomic integrity, right? So replication stress, what does replication stress means? If the DNA is undergoing replication super fast, when cell is hyperproliferating, you have very short window to complete your DNA replication. Yeah, there are actually different types of DNA polymerases that come into picture when cell is hyperproliferating. This is not our standard DNA polymerases 1 and 2, but there are certain various different isoforms of DNA replicases, and they are more error prone. They introduce more mismatches into replication. This is to ensure that the replication happens faster so that the cell can undergo cell cycle faster, but that introduces far more frequency of mismatch into DNA sequences. Now, these mismatches are repaired by MMR pathway, mismatch mediated repair pathway. There are various different external agents that introduce breaks into DNA. We all know that exposing your skin to excessive UV induces these DNA damage breaks, and we have UV related cancers being initiated, mostly in the places where there is high exposure of UV to the skin and less melanin uh, to 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 con- to protect these DNA damages, right? There are various different ionizing radiations we know, chemotherapy agents, which are now actually employed as a cancer treatment, introduce single strand breaks, right? So these single strand breaks are repaired by base excision repair, DNA damage repair pathway. These some of the agents can be even harsher and introduce double-stranded breaks, and these are repaired by homologous recombination or non-homologous end joining. Now, this kind of the non-homologous end joining is error-prone pathway. Again, it can introduce further errors into the system because we don't have a complementary strand to decide which strand to add. So these mismatches then are further repaired by mismatch-mediated repair pathway. So and certain products can get intercalated, which halts the DNA replication and leads to error prone introduction of bases, which are further there are further specialized pathway to repair these rare events of DNA damage. Now, this is a summary of various different these pathways and which checkpoints they hold during the cell cycle, right? As I said, most of the S2, G2, and G1 to S are held by checkpoints, right? So, non-homologous end joining, base pair excision, and what is NER? NER is the nucleotide excision repair. Yeah, it's between G1 to S. And there is only one checkpoint after mitosis, is the spindle checkpoint. Uh, there are the proteins that are involved in regulating these checkpoints, as all of us know, are the most critical proteins that are known in cancer initiation, right? P53. So, P53 is a major protein that regulates the important checkpoints G1 to S and G2 to M. And we all know 80% of the cancers are initiated when P53 is lost at germline level, at genetic level, right? So any mutations in P53, anybody has anything to say? I'll just go ahead. So any mutation, any loss of P53 is the fundamental reason. And as you can see, loss of P53 is going to release these checkpoints, right? So cell can transfer from G1 to S without correcting any of the DNA damage. Cell can transfer from G2 to mitosis without correcting any of the DNA repair. So these mutations get carry forward. Some of the mutations that lead further to cancer initiation, progression, get fixed as these particular cells hyperproliferate and continue to grow. So these are called clonal propagation, right? So any mutation that are beneficial for cell growth and proliferation get fixed because these clones keep on going further and further and occupy most of the cancer tissue that is being developing in the system. And today we know that you do need 80% of the tumors need p53 loss by any means, as we'll talk about further, for cancer initiation. But that's not sufficient for cancer to grow further. What happens after p53 is lost? Many mutations start accumulating. Some of the mutation, like survival of the fittest, some of the mutations help cell grow further, help say cell utilize the available energy, available glucose, available ATP in better ways, and those cell populations continue to grow as clonal population. So in a given tumor, we can have many clones of various different somatic mutations. Yeah, these are called somatic mutations because these are not germline cells. These are somatic cells in your skin, in your breast, in your pancreas, in your liver. These are somatic cells, right? So as the checkpoint is released, these mutations that occur in somatic cell get fixed. So in a given tumor, in any cancer system, you find a clonal heterogeneity. Yeah, with various different somatic mutations being generated because of loss of these checkpoint inhibitors, right? P53 is one of the major ones. 80% of the cancers have to have loss of p53 for cancer to initiate. So this is just a summary that we talked about. We don't have to go into detail. So this is how p53 works, right? So how p53 is a checkpoint to make sure all the errors in DNA replication are corrected. And how does p53 do it? So these are two different scenarios, right? So we have a cell where p53 function is normal, and we have a cell where there is a mutation or loss of p53 where p53 cannot have its normal function, right? So now having loss of p53 is not sufficient. There has to be a DNA damage through some external mechanisms or due to hyperproliferation has to be induced, yeah, for p53 function to come into place, right? So once DNA damage occurs, or for any other means, p53 is activated either through DNA damage or through hypoxia. P53 binds to DNA, right? Once p53 binds to DNA, it activates the transcriptional machinery, which gives the checkpoint inhibitor expression p53, GAD55. These are the ones are involved in actual DNA repair, right? If DNA repair is successful, cell divides and goes on to further life. But if DNA is not repaired completely, the apoptotic gene cycle gets activated and the cell is sent into apoptosis. Now, when p53 is not present, is lost in the system, after DNA damage, there is no p53 dependent gene activation, there is no checkpoint inhibitor activated. These mutations start clonally propagating and get fixed into a growing clone of cells population, right? So again, something to iterate here, having p53 lost itself is not sufficient for you to grow into a malignant tissue. You need to have a secondary event, DNA damage or something else, external factor, to grow. And that is the reason, even if you have germline p53 mutant, until old age, you don't show any symptoms of cancer. Is when the cell machinery or the more and more DNA damage is accumulated, the secondary effects start showing, right? And there are beautiful papers, actually, if you are interested, I can share. Nowadays, people do mouse studies with intravital microscopy. So they put up a tiny microscope inside a mouse mammary gland. So they have a wild type mouse and p53 mutant mouse, and they look at how the breast ducts, the milk producing ducts, grow and shrink. So during a life cycle of a female, these ducts grow, proliferate, and shrink back, right, every month. The mouse mammary menstrual cycle is about 2 weeks, 10 days to 2 weeks. So this growth and shrinking happens on a regular basis. in both p53 mutant and wild type mice. But in p53 mutant, the hyperproliferation rate is quite dramatically increased um, because the checkpoint inhibitor is lost. And still, that is not sufficient for the tumor to form. Yeah, so there is no tumor formation in p53 mutant mouse despite showing hyperproliferation in the cells. It's the surrounding immune system that contributes or that decides whether these hyperproliferating cells are going to be converted into mutant or not. Because what happens, this is still a simplistic picture, when you do have mutant cells, these mutations express certain immune, let's call them immune checkpoints on the cell surface. These altered immune proteins call in immune system to kill these cells, right? So majority of these mutant cells, when exposed to the microenvironment of or the stroma of the tissue, are going to be attacked by our host immune system. So only when these hyperproliferating cells in go start expressing the cell surface molecules that are similar, that are not recognized by our immune system, it can evade, it can bypass the immune system attack and grow further. Yeah. So as of today, even though this slide is actually very simplistic picture, there are multiple factors that play into for anything going wrong into developing a tumor. Yeah, so just having DNA damage and p53 loss is also not sufficient for you to develop a tumor. And what we say that we see increased rate of cancer development and tumor cases in today's world is that tells you how many different factors are actually coming together to initiate these cancer process. So when you see one cancer, despite increasing rate, there are multiple cancers that were initiated in normal body that were killed down either by p53 or by our immune system, right? So the ones that we see outperforming, growing, are actually the rare events in our human system, cellular system. Yeah. So another factor that comes into play is along with p53 checkpoints, the actual proteins that are involved in repair, right? And all of us know BRCA1, BRCA2 proteins are part of the checkpoint and DNA replication machinery, mostly involved in mismatch repair and homologous recombination. Along with p53, there are certain cancers, albeit to less frequency, require BRCA1 to 2 mutation. So about 20% of breast cancers are with BRCA1-2 mutation. About 80% of the breast cancers always have p53 mutation. So if you have a p53 mutation, there is about 90% chance that you will develop a cancer, any human system. But if you have BRCA1, BRCA2 mutation, there is about 80% chance that you're going to develop cancer in your lifetime, right? And BRCA2, BRCA1 mostly are involved in two major DNA repair pathways that are employed during after DNA synthesis phase. Yeah, homologous recombination and mismatch repair. So this is just a summary of what we talked. Now, how do we use this system that has gone wrong in DNA replication to our advantage to develop therapy against, right? So in a cancer hyperproliferative state, DNA is replicating faster. It is introducing more and more mutation. It is bypassing the DNA damage repair pathway. Can we employ these systems that are gone wrong to develop therapies against, right? So if we can prevent the DNA damage response in a hyperproliferative state, we can prevent the tumor formation. Yeah. So the DNA damage is repaired, the mutation is fixed, and the cell proliferates further. If we prevent this DNA damage repair in a cell that has gone wrong, we are going to prevent fixing the mutations, and eventually the damaged cell will undergo apoptosis, right? So either we prevent cancer formation, or once the cancer is formed, we target that mutation, that whatever the mutation that is fixed, and suppress further tumor progression. Cancer prevention and cancer therapy, right? So one of the major successful drug that has been developed. Now people have been trying targeting p53 multiple different ways. People have been invested in developing drug against p53 for 20 odd so years. There are at least 500 labs invested in working with p53. But p53 is a very fundamental protein and elusive to work with for drug therapies. But what people have become successful is in targeting BRCA1 and BRCA2 related cancers, mutation related cancers, right? So, do I have a better slide here? I don't. So when BRCA1, BRCA2 is recruited at the DNA repair pathway, at the DNA damage, BRCA1, BRCA2 employ the DNA damage repair machinery, and BRCA1, BRCA2 will call in PARP1 to the damaged part and repair the DNA damage, and the cell will get fixed with the mutation and undergo further proliferation. Now, in the absence of functional BRCA1, BRCA2, PARP1 is what is a major factor that takes over to bind the DNA. Now, in the absence of BRCA1, BRCA2, or in presence of BRCA1, BRCA2 mutation, which is non-functional BRCA1, people have developed PARP inhibitor to be administered to patients. Now, PARP inhibitor will block binding of PARP to damaged DNA. And when there is no DNA repair, the cell, instead of going DNA damage repair pathway, will undergo apoptosis. So in presence of BRCA1, BRCA2 mutation, when PARP inhibitor is administered to the patients, most of the tumor cells will undergo cell death. Yeah, and that's how the tumor progression is halted. So olaparib is the name of the drug which works as PARP inhibitor, and that is the first line therapy now administered to all BRCA1, BRCA2 mutations associated cancers that have developed. But again, these are only 20% of all the cancers, because BRCA1, BRCA2 mutation happens only in 20% of the cancers, right? We still so far do not have any successful drug to have similar mechanism employed to bypass p53 mutation related DNA damage bypassing. Yeah. So let's go to the next step in central dogma, which is transcription. Now it appears simple, but it consists of various different steps within itself, right? So you have initiation of transcription mediated by RNA polymerase 2, along with various different transcription factors that decide at which promoter region to bind, which promoter region to bring in polymerase 2 and initiate the transcription. Right now, depending on what kind of gene it is, how long the gene is, not entire mRNA is made in one go, right? It can be initiated and paused. One of the major cell system where pausing of the transcription is used is in a oocyte development, right? So before fertilization, most of the transcriptome of an oocyte is in a paused state. This is because to save energy to complete the transcription immediately after fertilization. So immediately after fertilization is a trigger. The paused transcription is released. All the transcripts are made, and the transcripts are ready to translate into protein and take the fertilized embryo into zygote. Yeah, because that's the first initial few replications are the ones that are happening at the fast pace in after fertilization. So this is to have all the proteins in a prime activated position, all the transcripts. Most of the transcriptome in oocyte is in a pause state. So pause is generally released, activated during the elongation phase of transcript splicing happens, post-processing happens, and then we have a mature mRNA that gets translated, that gets transported out of the nucleus and then gets translated, right? So splicing, just a recap if people don't know or have forgotten, that the mRNA that is transcribed from DNA has exons and introns. For it to be able to translate into a functional protein, we need to get rid of these introns through splicing. This splicing is taken care by a huge machinery called spliceosome, which we'll talk about in a next slide. Now, why this splicing is important? Because huge proteins like p53 or even BRCA. BRCA is amazingly complex for splice variants. If any of you are interested, you can look at the splice variants of BRCA. BRCA1 and BRCA2 both, and there are multiple versions of BRCA functional, and same is the story for P53. There are multiple exons, introns, and multiple combinations lead to various different aberrations and various different functions. The complexity itself is huge. Anybody's interested, I can share these, and you can study various different functions of various different isoforms, right? So, I had one slide here about spliceosome, which I don't see. But so to prevent these various different splice variants which are gone abnormal is mostly through the aberrant splicing function of a spliceosome. So if you target the mutated spliceosome or the spliceosome that's gone out of function, you can rescue the P53 aberrant splicing mechanism, right? So people have attempted rescuing P53 function by targeting these spliceosome associated mutations, right? Now spliceosomes have tissue specificity. There are various different tissue specific components or the binding partners in a spliceosome, and that's what this slide is summarizing about. These are the ones like PTBP1 is bladder specific, SRF1 is a breast specific or cervix specific. Various different components have predominance in a spliceosome in various different tissue targets. And these particular proteins, if they are targeted, they affect various different functions associated with various different fundamental cell related functions that contribute to cancer formation. So once the splicing is done, we have certain drugs that target spliceosome, but they still also have not taken up as much because of the tissue specificity that we mentioned about. The next step that we employ for developing cancer therapy is actually the expression, gene expression from, gene expression machinery from the DNA sequence, the gene sequence, right? The gene sequence is promoter, we have enhancers and stuff, and the entire coding region that is transcribed into mRNA, which is further then spliced through joining the exons depending on what particular function, what particular splice variant is important. So again, the regulation of gene, while transcription and translation is quite complex, right? So there are various different machineries that are employed in making gene expression successful, and anything that goes wrong in any of these systems can lead to aberrant gene expression. So the very first control on gene expression is at your 5' UTR, where the transcription factors bind, right? So any mutation in the transcription factor or any mutation in the promoter region can lead to aberrant transcription. Now we talked about fusion genes in last lecture. About, I gave you one example about BCL and RUNX1 fusion genes, right? So RUNX1 has inhibitory domain which is removed in the fusion gene, and RUNX1 binds many of the gene targets that are not its bona fide targets. So any aberrant mutation either in the transcription factor or promoter region will completely give away the normal transcription mechanism, right? The splicing that we talked about is dependent on the specific sequence at the boundary of intron and exon. Yeah, any mutation that happened during these boundaries will throw off a splicing machinery, right? So giving rise to aberrant transcripts, which will give rise to mutated and abnormal proteins further down, right? The poly A tail sequence and the termination codon at the end is critical to know for a RNA polymerase 2 that this is the place we stop transcription. Any mutation in these systems will either lead to truncated protein or extended protein, which is going to cause again havoc into a translated protein system. Now, there are many of the complex genes have 3' UTRs, which further employ regulatory elements like snRNAs, microRNAs, which control where transcription gets halted and paused, or whether transcription is initiated or not. Right? So these regulatory sequences are embedded in the DNA sequence, and these DNA sequences then control or regulate binding of various different protein components that are required to complete the transcription, right? So we have transcription factors, we have enhancer binding proteins, we have co-activators that either enhance the transcription or suppress the transcription factors, RNA polymerase 2, and then there are spliceosomes, and there are protein mRNA, the way we have 3' UTR snRNAs and microRNAs binding 3' UTR. There are various different regulatory proteins that bind 3' UTR, regulating the transcription. So the complexity is further gone up with these proteins coming into play, and any aberrations in these proteins also will lead to faulty transcription, right? The next level of regulation of genes is beyond the gene structure.